guys, Mr. Anderson here. Welcome to Biology Essentials video 41, and this is mitosis, meiosis, and differences between the two. Most of this podcast will be on the steps of each division. Let's start from the beginning. A chromosome is a thread-like object made of material called chromatin. Chromatin is made of DNA and special structural proteins called histones. Chromosomes are stored in the nuclei of cells. You can see that a chromosome may be scrunched up into a very small package in order to fit inside of a nucleus. The basic construction of chromosomes and structure is the same in all of animals. The difference is that each species has its own set of chromosomes. The total number, for instance, all human cells have 46 chromosomes. The total number of chromosomes in the gametes of a particular species is referred to as the haploid number of that species. In our model, the beads represent individual genes, and the entire string of beads represents a chromosome. The magnetic centers represent the centromere. The purpose of mitosis is to make more diploid cells. It works by copying each chromosome and then separating the copies to different sides of the cell. That way, when the cell divides down the middle, each new cell gets its own copy of each chromosome. Before mitosis, the cell goes through interphase where the DNA strand of chromosome is copied, and this copied strand is attached to the original strand at a spot called the centromere, forming two sister chromatids. The string around the chromosome is the nucleus. Mitosis begins with prophase, where the sister chromatids condense into tight packages from the loosely bound DNA during interphase. Following prophase is metaphase, where each chromosome lines up in a single file line at the center of the cell. By this point in time, the membrane enclosing the nucleus has dissolved, and mitotic spindles have attached themselves to each chromatid in all of the chromosomes in the nucleus. Next is anaphase, where the mitotic spindles, represented by the strings of the inside of the nucleus, pry each chromatid apart from its copy and drag them to the opposite sides of the cell. The four sets of sister chromatids become two groups of four single chromatids. In the final phase, telophase, membranes form around the two new groups of chromosomes, and the mitotic spindles that provide the power to create these groups are disassembled. The cell then splits into two by a process called cytokinesis, creating two clones of the original cell, each with four single chromatids. In an animal cell, the visible division during cytokinesis is called a cleavage furrow, while in plants it is called a cell plate. Now, on to meiosis. The purpose of meiosis is to make haploid gametes, where mitosis makes a new cell with the same number of chromosomes. Meiosis results in a cell with fewer, half as many chromosomes. Meiosis is split into two separate parts called meiosis 1 and meiosis 2. Meiosis 1 starts with the copying of chromosomes and their condensation into compact forms, just like mitosis. In prophase 1 of meiosis, the replicated homologous pair of chromosomes comes together in a process called synapsis, and sections of the chromosomes are exchanged. The metaphase of meiosis 1 is different, though instead of lining up in a single file, the sister chromatids line up 2 by 2. These groups, called homologous chromosomes, are what separate during the anaphase of meiosis 1. If we look at the anaphase of meiosis 1 with a diploid number of 4, the result is two groups of two sister chromatids, rather than two groups of 4 single chromatids. This difference in chromosome numbers in the post-anaphase groupings is really the only big difference between meiosis 1 and mitosis. Membranes form around the two groups during telophase, and then the cell splits down the middle, creating two non-clones. Each clone has half the number of chromosomes as the initial cell. Meiosis 2 applies the process of mitosis to the two cells created by meiosis 1. Since the chromosome already exists in sister chromatids, interphase is skipped. The result is 4 cells called gametes, each with 2 single chromatids. Now let's review what we've learned. Let's take a look at mitosis. Now, meiosis. Thank you, and I hope that was helpful.